Hello, everyone. Welcome back. Thank you for joining us. Good afternoon. I'm Steve.
And I'm Scott.
Together, we're Backyard Musings.
And we are in Apple Valley. I am, and Steve is still in the secret bunker, sort of secret bunker in Utah. And we are talking about a health issue today. So pay attention. We like these kind of stories because they really do help us, and it's pretty much common sense here.
Just before we jump in, just so you know, between Christmas and New Year's, we're just going to publish, like we did last year and the year before, one segment per day. That you're probably going to be with your family, and we're going to be with our family, and so one a day probably. That's going to be good. And then after New Year's, we'll go back to the two a day, which you love. Okay.
Yes, they do.
A fatty diet doesn't just damage the liver, it rewires its cells in ways that give cancer a dangerous head start. Eating a diet high in fat is one of the strongest known risk factors for liver cancer. So when I'm thinking about this, like avocados have fat, but that's a good fat, isn't
Good
it?
fat.
As compared to like the fat on bacon.
Yep. Salmon, fish.
Oh
good,
yeah.
Good fat, good oil. So
But I like to cook with bacon grease. It makes it taste so good though.
yeah, that's
Once in a while, is that okay?
well, what's once in a while?
Like every other
Once in
day,
the morning?
every third day.
Once in the morning?
New research from MIT explains why, showing that fatty diets can fundamentally change how liver cells behave in ways that make cancer more likely to develop. Would you like to know more?
The study published in the journal Cell, same study we referred to earlier today, on December 22nd, 2025, found that when the liver is exposed to a high-fat diet, mature liver cells called hepatocytes, undergo a striking shift. Instead of maintaining their specialized roles, these cells revert to a more primitive stem cell-like state. While this transformation helps the cells cope with the ongoing stress caused by excess fat, it also leaves them far more vulnerable to becoming cancerous over time. Not good.
Good, yeah. Pinch poke, you owe me a coke. Quote, if cells are forced to deal with a stressor, such as a high-fat diet over and over again, they will do things that will help them survive, but at the risk of increased susceptibility to tumorigenesis, says Alex K. Shallak, director of the Institute for Medical Engineering and Sciences, the J.W. Kiekhaefer Professor in IMES, and the Department of Chemistry, and a member of the Koch Institute for Integrative Cancer Research at MIT. It goes on. The Reagan Institute at MGH, MIT, and Harvard, and the Broad Institute of MIT and Harvard. Current and future president of the United States. Let's just say
that guy's got a big business card. Okay. The team also pinpointed several transcription factors that appear to drive the cellular regression. Because these molecules help control whether liver cells stay mature or revert to an immature state, they may offer promising targets for future drugs aimed at reducing cancer risk in vulnerable patients.
High-fat diets are known to promote inflammation and fat buildup in the liver, leading to a condition called steatotic liver disease. This disorder can also result from other long-term metabolic stresses, including heavy alcohol use, and may progress to cirrhosis, liver failure, and eventually cancer.
Yeah, not good. Any of those three are not good. To better understand what drives this progression, the researchers focused on how liver cells respond at the genetic level when exposed to a high-fat diet, especially which genes are activated or shut down as damage accumulates over time.
So the team fed mice a high-fat diet we love mice, and used single-cell RNA sequencing to analyze liver cells at multiple stages of disease development. That makes sense, single-cell RNA sequencing. This approach allowed them to track changes in gene activity as the animals moved from early inflammation to tissue scarring and ultimately liver cancer.
Early in the process, hepatocytes began activating genes that promote survival under stress. These included genes that reduce the likelihood of cell death and encourage continued cell division. At the same time, genes essential for normal liver function, such as those involved in metabolism and protein secretion, were gradually switched off.
Some of these shifts occurred quickly while others developed more slowly. In particular, the decline in metabolic enzyme production unfolded over a longer period. By the end of the study, nearly all mice on a high-fat diet had developed liver cancer. Oh, not good.
Yeah, not good. According to the researchers, liver cells that revert to a less mature state appear to be especially susceptible to cancer if they later acquire, bless you, harmful mutations. Bless you. Quote, these cells have already turned on the same genes that they're going to need to become cancerous. They've already shifted away from the mature identity that would otherwise drag down their ability to proliferate. That's according to Zoonis. Oh, I don't know. Did we refer to him earlier?
Yeah.
Zoonis? Okay. Going on with the quote, once a cell picks up the wrong mutation, then it's really off to the races, and they've already gotten a bad head start on some of those hallmarks of cancer. So
yeah. The team also identified specific genes that help coordinate this shift back to an immature state. During the course of the study, a drug targeting one of these genes, thyroid hormone receptor, was approved to treat a severe form of steatotic liver disease known as MASH fibrosis, and that's an acronym just like the TV show MASH. In addition, a drug that activates another enzyme highlighted in the research, HMGCS2, is currently being tested in clinical trials for steatotic liver disease.
Another potential drug target identified by the researchers is a transcription factor called SOX4. This factor is typically active during fetal development and in only a limited number of adult tissues, but not the liver, making its reactivation in liver cells particularly notable.
After observing these effects in mice, the researchers examined whether the same patterns could be found in people. They analyzed liver tissue samples from patients at various stages of liver disease, including individuals who had not yet developed cancer.
The human data closely matched the findings in mice. Over time, genes required for healthy liver function declined, while genes linked to immature cell states became more active. Using these gene expression patterns, the researchers were also able to predict patient survival outcomes.
Well, that's good, right?
Yeah, I mentioned it before. My father-in-law passed away from liver cancer, complications of a medication that he was taking that caused the liver cancer to progress rapidly.
Oh,
and he didn't want to do anything about it, and they gave him a time limit of six months, and it was about 6 months. So sad deal.
While cancer developed within about a year of mice, the researchers believe the same process unfolds much more slowly in humans, potentially over a span of roughly 20 years. I guess that's because mice, their longevity is different from ours, so I guess that's on scale. The timeline likely varies depending on factors such as diet, alcohol use, and viral infections, all of which can encourage liver cells to revert to an immature state.
The researchers now plan to explore whether the cellular changes triggered by a high-fat diet can be reversed. Future studies will test whether returning to a healthier diet or using weight loss medications such as GLP-1 agonists can restore formal liver cell function. So that's the same GLP. That's that same rapid weight loss.
Yep. We've talked about that.
Going crazy.
Yep. They also hope to further evaluate the transcription factors identified in the study as possible drug targets to prevent damaged liver tissue from progressing to cancer. Quote, we now have all these new molecular targets and a better understanding of what is underlying the biology, which could give us new angles to improve outcomes for patients, Shalek says. Love it.
The drug that affected my father-in-law was Lipitor.
Lipitor.
Yeah. And he was getting tested very regularly. And just the last test that he got was the long face test. It just wasn't good. So
wow, watch out for Lipitor then.
Yeah. Just be careful what you ingest. So anyway.
All right, folks, thanks for joining us. Catch you tomorrow.
Take care, everyone. We'll have a good night.